Before that, just a bit of introduction. Hi, my name is Bahia. I'm a master's student taking Bioinformatics and System Biology in Institute of Bioscience. Basically, Dr. Fiza is my supervisor. So today I'll be helping her on explaining a bit more tips and tricks of bioinformatics and such. Okay, I'm just gonna share my screen. Can you guys see my screen?
Yes.
Yes, okay. Thank you, Fatia. I'm just gonna ask for permission to stop my video so that it doesn't, the internet doesn't lag. So for that, okay. So for today's practical, this will be your first practical under the course of DSM Practical Course 4301. In this practical, basically, you'll be learning on how to access information from databases out there. Most databases, there's free databases, online, offline. There's like so much type of databases. And for this practical per se, you'll be basically by the end of the day, we expect you guys to know how to access literature related to anything actually, to a topic, to a concept, to a theory, basically to your target DNA or even protein of interest. And then secondly is on how to retrieve DNA and protein of interest information from databases. I'll give you some tips and tricks on like where to find which database to go to, and this kind of stuff. Because sometimes we have to pay for it just to access the database. But most of the time, there's free databases that have been widely used, and I would say used as a standard around the globe. So it's better to go to the free one rather than the paid one. This practical is divided into 7 parts. If you look at the practical manual beforehand, we'll touch on how to find, basically how to navigate around NCBI PubMed, and then how to navigate around NCBI nucleotide collection. And then last part is how to navigate around UniProt, which is a protein database. So without further ado, so for the first point, 1.1 practical sections is to find out about a protein by its name. If you guys have any question, just raise your hand or just speak it up. You do not have to wait till the end of the practical. It's okay, I won't mind. So if you would like to write a literature review on a strange term called cytotypes, never heard about it before as well, so that you have never heard previously, where do you go from that? How do you find out more about this familiar obscure term with a strange spelling? So you can opt for Wikipedia, but I mean just for a glance, for a skim through, but I would really, I mean, everyone would really suggest go to NCBI PubMed on the website, on the browser. The only difference is that if you write NCBI, okay, if you just write NCBI, this will be the interface of just NCBI, which you guys will see a welcome to NCBI here, and then like some buttons around here, and then like this one is the major thing, a drop down menu button, which shows all the collections under NCBI. NCBI is like a library. Under this one library, there's a lot of databases, there's a lot of collections, library collections basically. So these are all the collections that basically collects all informations accordingly. So it says here to go to PubMed. So you can just either click PubMed here and then type in cyclotide, just like this, or you can write down NCBI PubMed in the search box and then just click on PubMed. This will be the interface for NCBI PubMed per se. It's just the NCBI. So it says here, type in cyclotide in the search column and click search button. This will be the sorry. This will be the search. Basically the output search results that you get if you type in cyclotypes. Different people will probably have different like random searching. Um, Here's just the filters button that you can filter according what you want. Either okay, let's me click this one. It's not supposed to be there. Okay, now change. So according to the text ability, article attributes, article types, whether you want it to be a meta-analysis, clinical trial to say, just a review or just some books and documentation. You can also choose the publication date, either 1 year before, 5 years, 10 years, and the custom range from 2021, sorry, 20 for 1997 to 2021, say, and then the journal category and some additional features right here. And then there's a timeline. There's a few other buttons here that I'll explain more as we go. So it says here from the format drop-down menu button, select abstract. As you guys see here, it only consists the title, the name of the authors, and the basically all the information needed of that um, publication. And a bit of abstract, but it's not a full abstract. So what you can do here is to click on display option rather than click on summary, click on abstract, and then it will change into a full abstract with all related information. So rather than have to click here and open a new tab just to read the abstract, you can just change the display option here and then click on abstract rather than summary, and then you get the abstract in the first glance, and then as you go you'll find more abstract as well. So that's a good thing rather than having to open up into a new tab. You can just click here. So for any entries in the result list, click one associated author names to display all the literature of the author. Let's see, we wanted to click. Let's just click this Chindandu. This guy would probably pop out. I mean, if there's lesser guy with the same name, um, the author would probably pop up every single time it's related to cyclotide or something. Just like Dr. Nurufiza's name if we search for viruses, basically chicken viruses. Because every researchers have their like own niche research field. So most often than not, their name will pop up again and again and again in the same research niche field. So once we do this, uh, we're just going like a random searching right now. So let's say you wanted the first one and the second one. What do you like? If you want to get this information, what do? Do you like click here and then copy it and then paste it in a notepad or something else? But what you actually can do from just the button option provided by NCBI format is if you want to save the summary file, just click send to from the drop down menu button and then select file, save result text in summary format and then create file. What you can do is just click send to and then click as a okay. You can either click as send to or an email or save. I would rather choose save. This is an old, not old, but quite old interface of NCBI. Hence, they say that that's a drop-down menu button. But these are the quite fairly new. It's much more minimalist than ever. So just click on save, and then you can either choose all results on this page or all the results, which is just 2488 results just for this author per se. And then, or you can just click on what you select and then just click on the summary text or abstract text. This one, we just want the summary one, and then just create file, and then we automatically download it into your local host in the download file in a notepad basically. Okay, so this is the example once you save everything and once you choose and then save everything. Why isn't there any abstract right here? Because we opt for summary rather than abstract. But if you want abstract along the way, you can just click on abstract and then just save it likewise. So now we're done with the first. hope I'm not going too fast. At least if I'm fast, you guys could still catch up. Okay, so moving on to the second bit, which is to search PubMed using author's name. Let's say your friend has told you a very interesting paper on cyclotide, which was published by a group of researchers from the University of Queensland. He only remembered one name of the authors among the authors, which is great. Using PubMed, take the following steps to track down the illicit reference. Again, we go to NCBI PubMed. PubMed stands for publication something. So it's a collection of publications being reported out there. Yeah. Okay. So type in Craig, which is the name of your prospective author, in the search column and then click search button. You can just okay. And then you'll get this information, but bear in mind that we might share the same name. There might be like 10 authors with the same family name, just Craig, right? So what you can do is you might want to specify your search. So how do you specify? By adding more terms related to what you're trying to find, like Craig and then cyclotron and then uh, myself, as you go along, as you add more and more terms, you might lose some important search result. But it will try to specify your finding. But don't be too specific because sometimes there might be like the wrong terms that we use. So bear in mind, just go like add some and then remove some. So if this is the answer that you wanted, let's say these are the things that the basically the interesting paper that your friend has told you about, you can just simply click here if you do not want to read on this one. Let's say this is just the rule claim. So if you do not want to read more on this, you can simply, like previously mentioned, you can just click on abstract and then it will show you the whole abstract of it. So it's pretty easy because in abstract there's background, there's aim, objective, um, results and conclusion. So you get everything in the first glance in abstract. Okay. So this is such an example on how you could play around on what to display in your search result in NCBI. You click the title of one paper result, list again another one to certificate about. Save the abstract using the above command but select abstract text instead of summary text. So previously, I showed you on how to save the summary text. I'm just going to click on one. Okay, now it says here to try and save in the abstract text and see what will get. Mm-hmm. Saving for note. Wait, I'm a chat. Uh, try to, okay. So this is the abstract. So it says it stores all the information of the title, basically the information of the publicated journals. The name of the authors, authors information, basically the affiliation where these authors are from, and then the abstract of the said paper that we choose, and then all sorts of information. You will always save your information in, I mean, basically, you will always download all the information in a notepad. It's like the default thing for saving from NCBI. Why notepad and why not words? I would assure you that notepad, I mean, like saving information in notepad, saving text in notepad, will not use much of your space as compared to using words. But the only cons of using notepad is that you can't highlight it. You can't bold it, you can't do an italic, you can underline it. You can just look, see, and read and stuff. But words, you can like play around with the text and then move around. You can edit it, you can highlight it. So it almost act as a uh, like a book, like a physical book that you can highlight it. So but it's gonna like use your space a lot more as compared to Notepad. If you do not want to edit your information, just store it in the Notepad, basically .txt. If you want to edit around, highlight around, just boost some words, you might want to save it in the in the Word, Microsoft Word or WPS or anything. Okay. Now we're done with the second bit. We'll move on to the third part, because the third part is also using PubMed. Yeah, and the fourth one as well. Okay, so the third part, a field restriction queries. I'll show you some information on that, but I'm gonna try and find it first, because I think they deleted the page, but I'll see what I can do. Great for dealing with author's name or subject terms that can be found in different fields. Field restricted queries are useful for locating experts in particular field in given location. So again, you go to PubMed, we still wanted to find some publicated journals, but basically journal publication. So in the window, enter cyclotype and then TIAP and then China and then AD. So what is this TIAB and AD? So basically, TIAB means that we're giving the instruction to PubMed to find the term cyclota in either the title or the abstract, and or the abstract. It needed to be in the title and the abstract. China basically is the for AD is the affiliation of, as this is just to, again, you can, previously I mentioned that you can add more and more terms just to specify your search so that you get less and lesser results. However, sometimes some terms share the same position, like the word China. You wanted the word China to be in the affiliations of the author rather than in the title or the abstract. Because if we just look for China, it'll give us information in wherever the word China is being popped out, either in the name of the author probably, or even the abstract or the title. But now we do not want that. We only want to wanted cyclotype to be in the title and or abstract, and then from China. Only the people, if the author's affiliation are from China, not all, but probably one of the authors is from China. So this is how you specify your search, other than adding more and more terms. And then you can click on search, and then it says here to select the Medline option for format. So what you can do as well, just to specify your search, click on the filters, and then click on the Medline, and then there you go, you get something like this. Now from we'll get a more and more specified results compared previously. There's roughly around, I think, 2488 results. We just look for cyclotide and break. Now that we choose the turn on the filter line, and then we really specify our search, not just by adding one more terms, but also by adding the what they call the field restricted queries. Our result will just be more and more specified, and that is you in finding related articles or papers that you might want to include in your study. Okay. So that's one of the way, other than like on how to like navigate around and see that PubMed. And then the fourth one is to search PubMed using limits. When you want to quickly find out about a basic of subject you know nothing about, like nothing, zero knowledge of it. You only heard about it, you don't know much about it. It is best not to read a single highly specialized articles. No, not good. A better bet is to stick with the review articles, where one expert summarizes the state of the art for you. So previously I mentioned that there's Wikipedia out there that will just summarize everything that you need to know. But bear in mind that although Wikipedia has references as well, but it's safer and like much more better to just use PubMed and look for literature review of your subject or concept or theory of interest, because it's like a one-stop center. As soon as you find about cyclotide, you see like tons of literature review of just cyclotide, like what, how, why, when, in like in one paper for you to read. So it's better to go that way. Let's say you wanted to find more information about COVID-19. You just type in COVID-19 or SARS-CoV-2 and then they provide you like lots of literature review just stating. It's not even an experiment, it's just like a combined reading into one paper. So it provide you more information on like what, how, why, when, like who got sick, what's the statistic and everything that you need to know about SARS-CoV-2. So it's already like it's definitely a better way to read, to know about a subject by reading like a very general paper. Start with a very general paper and then you go specific as you go. So point your browser to the PubMed again and then type something in the search column. We can do that. Okay. One in the search column and then now I mentioned previously that we can like navigate around the filter button and choose accordingly. You can choose books and documents, clinical trials, normally cancer, and meta-analysis. Meta-analysis combine more than one study. Randomized control trial. For literature review, just click on review and then you'll get what you need to know about cyclotides. Yes, these are quite general topic they covered. Yeah, just like cyclotide structures with a little help from X-ray crystallography. That's very nice and cute title. Okay. There's not much of information here, but when you click on filter, you can also click on the additional filters, in which in the additional filter it provides you more options on how to choose and specify your search. So there's article types. There's a lot more. Under clinical trial, the phase 1 up to phase 4. Clinical trials just for veterinary, even command, data sets, directory, all those sorts. And then species as well, you wanted human or other human. And then languages, some uploaded in Turkish, some uploaded in Japanese. They're basically those country with monoculture, which and only have like one language. They would probably uh, publish their papers in those language. And then also sex of the interest, and then journal, and then age. Let's say they conducted a study from of a child from 6 to 12, and you wanted to study the effect of cyclotides in 6 to 12 years old child. So you can simply click here and then just click on choose. So there's a lot more options than you think. I don't know when you use it, but it's there for you to use, basically. So it says here, choose review and the article type, choose title and the sort by to search the articles for which title type is central. So what you can do here again, you can simply display options and then just move around here. And then you can also click here either a best match, the most recent one. Always go for a most recent one because you don't want to be outdated. Research phases are quite fast, like what do you call that? A field, yeah. And then publication date, and then first author, such thing to be sorted. And then per page, and then either show snippet or not show anything. Okay, it says here choose title underscore by. I know that there's no such thing as title to choose no more. So do not be alarmed by that. Again, I'll say here, this is the fairly new interface set by NCBI. And what we're saying here in the manual is probably like the second last version of it. Okay. So now we're done with using PubMed. Okay, let's see. We're gonna now, rather than using PubMed, we're gonna try to use one of in the NCBI library as well, but under the collection of nucleotide rather than PubMed. We're done with trying to look for literature review. Now we wanted the real deal of genetics or the real deal of biomolecular. This is genetics. So what you can do here is click on NCBI. So for 5, 6, 7, you're gonna play around with sequences and protein information. For 1.5 is you try to retrieve a nucleotide sequence, which is a combination the sequence of A, T, C, G. So imagine that you want to retrieve DNA sequence to clone a DH-based gene of E. coli. The gene is normally divided into protein coding and non-coding regions as well. Obviously, all of these informations are normally written in the gene bank report. So where do we find this gene bank report? Click on NCBI as previously mentioned. In this drop-down menu button contains all sorts of collections under the NCBI library. So rather than previously we used for PubMed to find publication, if you go to SNP, you find the single nucleotide polymorphism. If you go for OMIM, it stores you information of mutated genes and proteins information. If you go to genome, it will store you information, all of the genes that can be found in, let's say, a chicken, uh, and then a lot more, like lots more. So, but for this one, you can just click around here and just try, like, just move around the collections and see what they provide. But now, to retrieve nucleotide sequence, click on nucleotide. Okay, so once you get that one, secondary nucleotide database instead of PubMed, and then enter the accession number. Okay, accession number is all information published or recorded has different accession number. Like they will not share the same accession number. If they do share the same accession number, it only means that they are exactly the same. It's two things, but exactly the same. So it's basically the same thing. It's that you get this information. So according to the gene bank, basically nucleotide is the name of the collection, but under nucleotide, all the accession number and such, we call it the gene bank results. Yeah, it's the genetic bank result. The gene bank result. This is the accession number of the mus musculus, the name of species, and then the DUTPase mRNA complete coding sequence. Okay. Once we click search, okay, scroll down to observe the gene bank record. Generally, a gene bank entry will consist of 4 main parts. The locus name, which is this is the locus name, and then which is an arbitrary identifier, is followed by a short definition line as a unique accession number, and then the reference sections. This is the reference sections, and then okay, in this reference sections, there's underneath it, there's the list of authors' name. Basically, these are the names, or basically these are the authors that recorded this sequence. The same people can't report the same, uh, like the same sequence can't be reported twice. So it can be only reported once. And for this sequence, it is reported by this author with the family name Khan. And Varanasi, Chu, Chi, and Reddy. Okay, so it is reported and in with the journal title Mouse Deepest Cloning Engineering Structural Analysis. Okay, once we're done with the reference and then there's feature sections. Okay, in the feature sections, we will provide you information. Okay, Going up again, in the locus, it says here that the length of the mRNA sequence is 732 base pair. It is linear and mRNA. So in the feature section, it will state from 1 to 732, basically the whole base pair sequence, what it is, what's the organism, what's the molecular type, either DNA, mRNA, microRNA, all sorts, and then some reference of the taxon, basically the mass musculus code basically for it. And then the coding sequence, which is from 91 to 579 base pair, is the DUTPs. That's how it will also give you the translated amino acid residues from this one. Okay. But bear in mind that you get the features information if you get lucky. Some authors out there, they only provide you the nucleotide sequence, but they do not provide you the features information at all. It's either they do not have the information on the features, or they simply do not want the world to know about these features. They just wanted to recorded it. No, they just wanted to be the first one to recorded it in the world, basically that. Because again, the same sequence would never be allowed to record it twice. Hence why there's only one accession number, and why I said that, or why I mentioned, if they have the same sequence, they're going to be the same thing. Even though a group found it in the year 1997 and another group found it in the year probably still 1997, but from two different part of the world, part of the globe, yeah. So by then, it's a race against time. Who, like which research group will be the first one to record such sequence? So now, what's your
Yes.
Sorry?
Yes.
Yes.
What was CDS again?
What's the what?
CDS. CDS.
Oh, CDS. Okay, sorry. This one is the coding sequence.
Oh, coding sequence. Okay, thank
Coding
you.
Okay, yeah. It's a coding sequence because when we talk about mRNA, it's just the exons, right? We do not want the introns. So along with the genes, there's introns and exons. Once the introns are being omitted, there's just the exons, and the exons are the coding sequence that can be found in mRNA. So this isn't the DNA sequence because there's probably some bit left over here that's supposed to be the position of, or basically the sequence of where introns is. But I will not go further than that. That's some those are some genetics and genomic stuff. Okay, so let's say you wanted this nucleotide sequence for your studies. So should you just copy it like this? I mean, it's not, I mean, you can, but it's not suggestible because you're gonna include all the line numbers as well. So what you can do is click on FASTA, and then what you'll get is, you click on FASTA, and then it provide you the nucleoside sequence without, basically the mRNA sequence without the number line. Basically all 732 or 723 base pair that provided by the authors. So you can simply like copy everything here and then paste it again in a notepad like I said previously, and then just save it. Do not omit the first bit right here because this bit is like very, very important because nucleotide there's only A, T, C, and G, and like all sorts of combination of it. You're gonna get overwhelmed because you won't be working with just one. Probably you won't be working with just one sequence. So as you go along, like you get overwhelmed on how much file that you get. So this information is the most important bit. Yep. Okay, so you can do that, or just like NCBI, because gene bank is still under NCBI, nucleotide is still under NCBI, you can simply click send to, and then if you want the whole complete record of the gene bank record, including the reference, including the authors, you can click complete record, but I rather click just for the coding sequence. and then click on the FASTA nucleotide, and then just create the file. And then save your information just like exactly the same as what we wanted it. If you wanted the protein sequence, amino acid residue sequence, click that, and then it will also provide you, it will also save it to your local host just like that. So it's pretty easy, but again, in the feature section, I already mentioned that the location of the protein is just from 91 to 579. It will not provide you information from 1 to 732. So that's basically it. That's how you navigate around NCBI and nucleotide per se, in nucleotide collection per se, to get and basically retrieve a nucleotide sequence. Okay, some exercise for you guys here. So I won't do it, but I'll read it for you. So copy the sequence sections of an gene bank entry and highlight ribosome binding site, protein coding statements, start and stop. So how do you do that? Click faster. I did that. Copy and paste the nucleotide sequence in a Word document. Previously, if you just click send to and then save and then download, it will definitely store in the .txt and save in a .txt. But as I mentioned, you can't edit it around. You can simply just write, read, write, and see. So if you wanted to highlight and all sorts, copy the whole sequence, including this information right here, and then paste it into a Word so that you can like highlight stuff. So find ATG as a start codon, and also before ATG are the RBS, which is the ribosome binding site. So as the coding sequence that start from ATG can be found in nucleotide 91 and then stop codon 579. You might want to start this one. Although we already know it's from 91 to 579 because we provided the information right here per se, 91 to 579, right? But you might want to, let's say if there's no features at all. So how do you find where is the start of the protein coding segments? So this is the way, find ATG and then look for the stop codon. We just do it manually. It's still a way, it's a manual way. Okay, so moving on to the 1.6. Now we're done with on how to look for nucleotide sequence. Let's see. Some people do not want to study about nucleotide; they just want to study about the protein sequence per se. Although gene bank records provide you this information, which is the amino acid residues, the protein sequence, but again, like I mentioned previously, not all provide information of such features. I'm gonna try if I can find on the session number. Oops, sorry. The session number that do not provide you. It's not it. Provide you the information. But just bear in mind that there's a very high possibility of you not having the features of you have to do with the protein transition quite manually and such. Okay, so now let's say you just wanted the protein sequence. You can also use previously, like I showed you, you can try you just copy it here. This is exactly the information that you want, or you can opt for another fun database. It's free. It's a bit colorful as compared to GenBank and NCBI, which is named as Unicode. Yep. So as compared to NCBI previously, NCBI used the minimalist theme of such, and while Unicode is a bit more colorful, but a lot more buttons to be clicked, which is definitely nice. And it's like a one-stop center for proteins. Do not come here for nucleotide sequence, okay? Do come here for proteins. So point your browser to Uniprot, type in bacteria lactobacillus in the search window of Uniprot KB and then click the search button. So Uniprot KB composed of two sections. That's the Swiss prod, and then which is being reviewed, and then under the tremble, which is so the Uniprot KB entry is made up of 10 parts. Okay, I'll go this later. But I'll mention names and origins, protein attributes, general annotation, anthologies, sequences, reference, cross references, entry information, relevant documents. So what's the difference between reviewed and unreviewed? Sometimes most people would opt for reviewed because it's more legit as compared to unreviewed, but not saying that the unreviewed is not good. It's just that reviewed is manually annotated that like it's there, it has been proven wet labs that is there, while unreviewed is computationally analyzed, not saying that it's not there, it's just that it's being approved because of an algorithm of Unicode. So basically that's it. But you can still use information from unreviewed. There's no such problem. It's just it depends on your options and your interest. Basically, that's it. Okay, so click on the entry P83002. Like previously, in GenBank record, there's a session number, right? Basically, a session number is like a registered number, the registered number for that information. So in Unicode, there's a session number as well, but it's not for is not the position is not the same. So a session number of Uniprot can't be found in GenBank and the GenBank can't be found in Uniprot because it's two different databases. So bear in mind of that. So you can just look around for this one, then click here. Mhm. So here, as mentioned, there's 10 parts. There's names, origins, the status of it, the protein attribute, basically the functions, the biological process, the keywords it's related to, the protein family name, and then the names and taxonomy of the organism it is found from, the subcellular location, where is it located in the cell, okay, and then some processing information, the molecular processing, amino acid modification, and then the 3D structure of the what is this protein. But okay, Uniprot gonna lag a bit, but it depends on your local host. It still works just as fine. Just like because it's not a lot of information and it's just it's just a moving 3D object, so it probably like use a lot more of your data and space in your host, hence why it's a bit slower as compared to using NCBI nucleotide. And hence why NCBI nucleotide collection just use a very minimalist um, option because it stores a lot more information as compared to Unipro. So it's a pros and cons kind of thing. Okay, this one, the name of the gene is MMF. So you are quite lucky because not every protein already has a 3D structure. But for MMF2 from bacterial Lactobacillus already published and recorded. So this is the 3D structure um, of your protein of interest. Not all protein already have a 3D structure. They are still trying to make a 3D out of it because it's not easy. This thing is built by computational biologists. It's one of the branch under bioinformatics. So I mean, it's fine and like very interesting. Just like you can move around. If you know how to interpret, if you know like what to do about it, it'll be fun. But if you do not know, just omit this information. Okay. But it's nice to know that okay, this is the bit. There's an alpha helix right here. There's a loop right here. So it's still information for you guys, right? Okay. So there's information of the family domains and then for the sequences. Okay. Previously mentioned that I said that do not come here for nucleotide sequence, but actually they do provide nucleotide sequence. But normally people don't go here just for nucleotide sequence. They want it solely for the amino acid residue sequence because you can go for gene bank for the nucleotide sequence. So why be here? Yeah. Okay. As you guys can see here, that's dash one, right? 38300 dash one, because this is the first variant. Normally, some proteins has more than one variant, although it's the same protein. So you'll get something like dash one, dash two, dash three, dash four if there's four variant. But for this protein per se, they only found one so far. So there's only one variant. They won't call it variant because there's just one, right? So it's that's just one sequence per se. Okay, and then there's information on the mass spectrometry, a bit of chemical things like that, and then cost reference. All this information that they provide in these result sections, where is it from, and all sorts of stuff which you can simply click on. Okay, this is just some information. For those who wanted to know more about 3D structure of proteins, there's this one database called Protein Data Bank. Yeah, it's called Protein Data Bank. It's a localized database for people who wanted to study more about the 3D structure of protein. So molecule of the month is the VEGF in angiogenesis. Fun facts. Okay, so moving on to this question. So what is the amino acid sequence of this protein, MM2 protein? This is the sequence, amino acid sequence. You get this information in GenBank as well. You just type in, but you can always just go to UniProt for a more holistic information that you can have. So, and then highlight the amino acid residues that form a covalent bond besides peptide bonds. So we'll go to the PTM or processing. This is protein translated modification or something. And then processing with it says here form a covalent bond beside peptide bonds. So these are the peptide bonds. The peptide bonds can be found throughout the whole protein, which makes sense. The one that who between the amino acids are peptide bonds. And then there's disulfide bonds as well that has been identified from the position of amino acid 9 to 14, which is disulfide bond is a part, not a part, but is a type of covalent bond. So the graphical view is says here, if this is 1 to 37, it's roughly around 9 to 14, which is somewhere one half before one half. So this is the highlighted amino acid residues. So if you want to highlight from 9 to 14, I never click on this one, but let's click on that one. Uh-huh. So it's between C to C. This bit is the disulfide bond. So that's how you get more information on just the protein. Because normally, I mean, like in a beginner level or something, you try just to understand how a protein works, right? But as you go by in protein studies, in the protein research field or something, they wanted to know the exact information or the exact interaction between one amino acid and another amino acids. Hence why it's a very, very detailed field. A bit of chemistry, a bit of physics, and computational biology, and basically that's how you get a 3D structure. Without the information on the interaction between amino acid, we wouldn't get a 3D structure. So we could kind of assume that the protein with no 3D structure is a protein that does not have info, that the world does not have information on the interaction between the amino acids. Because amino acids have different characteristics. Some are hydrophilic, some are hydrophobic, some are polar, non-polar. All those sorts of chemical characteristics provide you the information on how amino acid interacts. And when amino acid interact, it will give you a 3D structure. So yeah. Okay, so to get the FASTA format, just click on FASTA under the sequence sections. Then it will simply this bit. It does not have the sheets like NCBI in which we can just send to and then download it. This one you have to copy everything and then paste it into a notepad, and then you're good to go. Yeah, just like that, and then just do. Okay, the last bit of the practical. So now that you learn on how to um, like retrieve nucleotide sequence, you learn how to retrieve amino acid sequence, amino acid residue sequence. Now, let's say you want like a collection of, you wanted to study a system or you wanted to study a function. So there's probably more than one protein that you need to like understand and find function about it. So how to build your own specialized sequence data file using Unicode. So I'm just going to click here again. For it is by downloading a large collection of similar protein sequences that enables you to use it with other analysis programs. Because sometimes you wanted to do some last thing, you wanted to do some aligning, or you wanted to do some phylogenetics, more such. So it's pretty hard to like click and then click on the FASTA file and then download it, paste into a notepad or something. So what you can do is just I'm gonna do this again. Click on the search button. Search window in the Unicode. You can filter by reviewed. Let's say for this one you wanted just to get the information on the review paper. Okay, so you get 31 rather than previously I think around quite a thousand or something. And then once you click on the review, there's option for you to move around and whatever. Let's say you wanted the length to be like lower than 100, right? It's gonna take a while. It's quite heavy. Mm, okay. Basically, the length of all the proteins that can be found in bacteria, sin lactococcus is up to 993. Let's say just wanted to study on the protein that's lesser than 100 amino acid residues. So 100 is still too much, but at least you get something like 1, 2, 3, 4 example, but I'll just click it for integrity. Okay, let's say you want to, okay, here we go. Okay, no, not much. Okay. So once you're done deciding like whatever you decided to do, so okay, since I said that for my research, I probably wanted to study on about the protein in bacteriocin lactococcus, which has the length of amino acid residue lesser than 100. I do not care what's the name of the protein. I do not care the functions. I just care that as long as it's under bacteriocin lactococcus and the length is lower than is lesser than 100 so not less than 100 amino acid residue. So what I can do rather than I click one by one is there's 20 results out there. Is a lot. So what I can do is just I click on download button, and then I can click on the whole selected what I selected, and then I have the option to save it either in FASTA, type separated value in Excel, in text, or in list. I'm gonna try to download in text first and see preview first. Then let's see how will like the information that will get. Okay, this is the example of it. It shows you all the information here. Okay. As you go, I mean, like normally in the left hand side, you'll find a line number, right? But for this bit, it's not a line number, but rather the information the um, of the protein. So ID is the ID, AC is the accession number, and then like GN is the gene name, OS the organisms, OC is the organisms taxons, and then there's RX is the reference, RA is the reference author, RT is the reference title, another RT because it's quite long, and then RL is the reference literature review publication, journals results, all the sorts of information. Yep. Okay, this is FT is the features. It has elix, it has strands and all sorts right here. So it's, but I get it. I mean, some people they need this information, but if you find it overwhelming, you can do one by one. But let's say I do not want to do one by one, so I can do um, rather than click on text, because text will get you all the information as you can see in the search result as you click on it. Now let's try this one. What will I get? Okay, nice. I will just get the protein sequence, which is definitely nice. I do not care about the reference. I do not care about the features. I do not care where it is localized. I just wanted the protein sequence so that I can like maybe do an alignment after this. Yeah, to see if that's like a converged region or a conserved region or sorts. So these are the preview 10. So those are something what you see. And then you can either compress or uncompress. If just 20, uncompressed is fine. If it's 100 or something, you might want to compress first. Yeah, or it will take you a lot of time. Okay. Yeah, these are the informations that we get. You can just like copy here everything and then just mention why it's different. Or this is maybe like the old stuff. So, so that's basically on how you can navigate around Unicron and use all sorts of options out there. Just move around, click around. The buttons are free to use. And it's fun to like to see that there's a free and available database out there for you guys to use. Along the way in this bioinformatic like practical, you guys be using a lot of blast and then engine bank, accession number. You guys will see Unicode again. Also, so this is quite a good information for you guys on how to start bioinformatics. Yeah, bioinformatics is like a game. Uh, there's information out there, but there's no correct methods on how to solve the mystery. Some guy might just solely use Uniprot to find information of a protein. Some people might use another database of like still a protein database, but another database, not a Uniprot, to find more information about the same genes. But by the end of the day, they still get the same information. They are still trying to find the same gene, the same protein. They are still trying to look at the same experiment. So there's no correct way or there's no one best practice way on how to solve bioinformatics basically. As long as you get the answer by the end of the day. So a long way or a shorter way, all makes sense. So I guess we're done with the first practical. So I hope that me and Dr. Fiza, I hope that you guys know now that on how to assess literature review or any kind of literature out there. Um, related to your concept of interest, target of interest, protein, DNA, or even topic of interest, subject of interest, and then which is basically by using NCBI, PubMed per se. And then secondly, you guys are now able to basically have an idea at least on where to retrieve DNA and protein information from databases. Can either be NCBI nucleotide collection or NCBI, sorry, or you got this one. So yeah, so we're done with the first practical. Do you guys have any like questions or anything? The first practical is quite, I mean, like it's pretty general and easy and quite technical. So um, I mean, you guys can always go back here if you have more questions.
Okay, thank you, Bahia. You are done? Okay, Assalamualaikum and good morning, everyone. Okay, so basically, I think you uh, get some information, okay, about the public database. Okay, I asked Bahia, my student, postgraduate to assist you. Okay, so basically, it's also to train the student to become more familiar with what we call the bioinformatics. So this is the way. So you need to practice to make perfect, right? Okay. So do you have any question regarding this practical? Anyone? Can you follow? Zulaikha, are you there?
Yes, Doctor.
I can't hear.
Yes, Doctor.
Is that because my wait? Okay.
Doctor, can you hear me?
Yes.
Uh,
So are you okay? Yes.
yes, I can follow.
Okay. Good. All right. Okay. So basically I have upload the new action notes. Okay. Because the one that I put last time is not the latest one. Okay, so I have updated that one. And then you can also check your Putra Blast to see your groups for the practical, right? So you need to submit the practical every 2 weeks. So means your practical one is today, so you need to submit 2 weeks from now. All right. So for the next week practical, we will lecture and practical we will have face to face, right? And then during the practical, we have 3 hours. So maybe we will use half hour or 1 hour to discuss what we call practical one, the to the practical. If you have any issues while you did the practical, so you can just ask the question, okay, before we move to the next practical. Okay, all right. Okay, so I just want to summarize, okay, whatever that Bahia present or explain to you regarding the practical one. So let me share the screen. Bahia, can you let me share the screen? Okay.
You can now, yeah, can then you can now.
Okay.
I said to all participants.
Okay. Okay. So, class, can you see the what we call the website that I show here?
Yes.
Yes.
Okay. Right. Uh. Okay. If you remember that I have delivered to you the lecture on Tuesday. Right, so we talk about the databases, right? For bioinformatics, the database is very important, and then we have three common databases, and that is, I mean, worldwide use that databases. Okay, so this is the most famous one, or the most user friendly one. Okay, the NCBI. Okay, and then this one is by US, right? Okay, so from this database, you can actually explore so many things, okay, regarding the biological data, right? So as I mentioned last time, the what we call the definition of bioinformatics itself is to retrieve and to store, okay, and also to manipulate whatever data that we have, okay, or you can also get the information from others by using this bioinformatics, right? So if you see here the features of the NCBI itself, you have a submit here, download, analyze, research, develop. So this is something that in line with whatever that I have mentioned to you last time, right? And then if you go to here, okay, as I mentioned also, in the databases we have in the database we have a lot of another database. Okay, so the bioinformatics is arranged in such a way that make you what we call make your job or your work much more easier, because they arrange into what we call they organize into a very systematic way. So means that if you want to go only for the gene, you can go for the gene. If you want to go for genome, then you can go for genome. And then if you want to find out information about protein, so you can go straight away to the protein database. So it's a very easy to use, right? So please go back and explore, right? And then, as I mentioned, also, for example, you can relate with the current situation. Okay, the last 2 years, we are what we call by the SARS coronavirus 2, right? So of course, there is a lot information of the SARS coronavirus 2 will be submitted to this database, and then there's a lot of information that you can retrieve. Okay, so you can just click, okay, to play around. So this is the example of the coronavirus 2. So you can see the data about the nucleotide records, the clinical trial. This one is the PubMed, so it means all the information, the publication, the study, the experimental about the SARS coronavirus 2 are reported here. Okay, okay, so you can see a lot of literature that discuss and mention whatever progress that they have done in terms of to control or to or prevention from coronavirus 2, or even the treatment for the SARS coronavirus 2 like antivirus, something like that. Okay, so you can go and then read for this one. Okay, and then we have also interactive features for SARS coronavirus 2. So, for example, here you have the RefSeq, means the reference sequence for SARS coronavirus genome. Okay, so that if you want to go for the graphical display, so you can see here, you can view the genome of this coronavirus. Okay, okay, so you can see here. So this one is the what we call protein, or that we have a ORF1AB gene that encode for the protein for SARS coronavirus 2, for example. So you can just click, and then there's information. For example, here is the coding sequence YP, okay, here 00972 438189.1, right? So basically, this one is the ORF1AB polyprotein, right? So where's the location of this gene inside the what we call genome? Okay, inside the virus genome. It's free. Okay, so if you click this ORF1 gene, so it will divide into the small other protein also. Okay. Um, so you can read information. So it means that there's a leader protein. Okay, and then it has about protein length only about 180 amino acids, right? So you can see all the information here. You can play around. Okay, so for example, if you want to find out the information about the spike protein, so please go reach the spike protein. So this one is only of maintain, so you can go back. Okay. Okay, so this one is the S protein of the coronavirus. You see here the surface glycoprotein, okay, or the spike protein here, right? So how many length of the nucleotides? So they have about 3,000 nucleotide, and then the protein is about 1,200, right? So you can study for this spike protein. So from the which position, for example here, okay, the position from 13 to 304 position of this spike protein. So basically encode for the SARS coronavirus like protein. So this is the spike S1 NTD, right? Okay, so it's only contain about 292 amino acid, right? So you can find out information. Is there any important region in this SARS coronavirus 2, what we call spike protein? So, for example, like the what we call in the spike protein that we know that there are receptor binding domain, which is actually play what we call important role in the attachment of that protein to the host receptor like ACE2 in human, for example. Okay, so this is where's the receptor binding domain located. Okay, so you can play around and you can manipulate this region. Okay, for further research. Okay, for example, like if you want to clone this gene or maybe only this region, okay, to make what we call vaccine or whatsoever. So this is something, which is how do you find out the information and explore using the bioinformatics database, right? Okay, so it's all the information is here. Okay, right. So do you have any question? Is that okay? Okay, if you don't have any questions, so now please go to the practical, right? And then do your practical by your own. And then after that, you can discuss with the group to prepare the report and answer all the question inside the practical. All right. So I will give you, I will open this Zoom until 10:30. Sorry, 10:30. 11:30. Okay. So that you can do the practical and you can ask question if you do have any question. Okay. So Bahia will be here to assist you, right? If you want to ask anything. Okay. Do you have any other question?
Doctor,
Yes.
I'm not in any group. I just
Sorry.
registered new.
You just register.
Yes.
Okay. I will let you know later. Which group are you in? So
Okay.
this is the first time you register for this course or
No, this is
second
my
time?
first time.
Okay, so this is your first time. All right. Okay. It's okay. I will inform to the which class, sorry, which group they are you in. Okay, later on after this. Okay. So do you have any other question for the rest?
Doctor, about the
Yes.
lab report, how do you
Mm-hmm.
do the lab report? Like usually we have like abstract, and how do we do the lab report for this practical?
Okay, basically it's the same. You can talk about the introduction for the database, something like that. Okay, regarding the topic for the practical one, and then you can straight away go to the practical by what we call by the section. 1.1, something like that. And then the most important is to for you to do the for what we call for you to do the practical and then to find the answer or solution. So to do that you need to screenshot, okay, whatever step that you have done, okay, and then put it in your methodology inside the practical, and then for the results. Okay, so you can just mention whatever that you have find out. Okay, and then answer the question. And of course, in term of actually for the result and discussion, you can just do it in same what we call same session. All right, so that you can combine both result and discussion. So mention what is your result, and then you can discuss whatever that you think is related or is important for you to highlight in term of this practical. Okay.
So doctor have like any example the lab report?
Um. Basically, for this one, I think you can use the what we call similar template like any practical that you have attend before. As I mentioned, is the flow is the same. You have the introduction, and then introduction or abstract or summary. Introduction or abstract or background is normally it's the same thing. Okay, and then you have the methods. Okay, the methods in terms of the bioinformatic, you need to screenshot whatever step that you have done, and then for the result and discussion, you just combine, right? And then finally, answer your question, and then after that, you can add on the references. Okay, so this one will add on the value of your report. So means that if you answer the question, so you can justify to prove that your answer or your step or your whatever solution that you provide is support by another works, for either you find out from the journal or from the books, uh, uh, okay, as a source, then it's good that you can put in also. Okay.
Okay, doctor.
Okay.
Hello, doctor.
Yes.
What Nadira actually meant was, like, do you have an example report that you can give us, just for us to refer to before, starting to write the reports? Because this is quite new to us using software.
Yeah, as I mentioned, actually for the lab report is same for all. Okay,
Oh,
just
so like,
how the layout is the same. Just that the methods. Okay, the difference is the method, because the method, if you use the normal practical, the wet lab, okay, you need to what we call mention whatever step that you handle during the wet lab, right? But for this bioinformatic, you need to screenshot whatever step that you did. Okay, because you don't have any hands-on, I mean, practical, okay, doing any equipment or apparatus, something like that. So for this one, you need to screenshot the step, whatever step that you have done, and then you just screenshot. Okay, so this to show you that, or to prove that you have done for that part.
So there will be no discussion and conclusion like that lah, doctor.
As I mentioned, you can combine for the result and discussion. You can combine, means you find out that, okay, this is the result. So maybe you think that, for example, like let's say, if for example, like if you search what we call one keyword, for example, like SARS coronavirus 2, and then it shows you the a lot of answers. So which answer that you will choose, or maybe you can explain why, something like that. Okay, whatever, or maybe also you can also discuss about the question. Okay, so maybe that in the question it's ask you how many length of the amino acid that you find out, something like that, and then you can just uh, uh, what we call mention your result, and then just discuss a bit. Okay, why you choose? Why do you think that this only 200 best pair, for example? Okay, because of the position, something like that.
Okay, thank you, doctor.
Okay. So,
Doctor,
yep.
regarding
Yes.
protein or alters them too based on our choice ke?
Which one?
Like practical one, right? Like we
Mm-hmm.
have to find about protein or by author's name too by our own choice ke?
Which practical 1 point?
1.1 and 1.2.
1.2. Okay. All right. So basically here, the example of the protein that you need to find out the information is the phosphotide, right? Okay. So for the in the lab report, okay, you can follow uh what we call whatever protein or gene that we are suggested in the practical. But if you want to try with another protein, it also can.
Okay, thank you, doctor.
Okay. Because the aim of this practical is to make sure that you are familiar with the bioinformatic tools and also the bioinformatic database. Okay, so if you can use the same method to find out the different protein or to find out the different gene, so it will be good also. So it will shows that you understand about the process, and then you understand about the aim of the bioinformatic. Okay, so means you achieve that aim. All right. Okay, so any other question? No. Okay, so if no question, then you can proceed with the practical. So I will end the class at 11:13. So during that this 1 hour, so you can just ask question about the practical if you do have. Okay. Right. Thank you, everyone.
Thank you, Doctor.
Thank you,
Doctor.
Doctor.